Thank you all for being here. We really appreciate you coming, and I'm really excited to kick off the keynote speech of the annual Plant Biology Initiative Symposium. So this is, for those that are not familiar, this is an annual symposium that the plant biology group here at Harvard University puts on. We rotate around topics each year, and this year's topic is root microbial interactions in a changing world. Before I introduce our speaker for the evening, I want just a couple of quick announcements. The bulk of the talks are going to be in an all-day affair tomorrow. If you like what you see tonight, don't want to miss it. It is going to be out at the Arnold Arboretum at the Weld Hill Research Facility. It starts at 9 and will go much of the day. If you have signed up for the shuttle, then that will leave from the herbarium at 8 a.m. And then bring you back at 6 or something like that right after the reception. And there are also going to be a bunch of wonderful posters from students and postdocs within OEB and surrounding universities, so it's going to be a great affair. Other announcements. Note that the parking at the Arboretum is free, but it is first come, first serve, and it is limited, so keep that in mind. There is some parking around in the area on Center Street and Bussey Street and some areas right there if you need. Okay, I think that's all I've got on announcements. Okay, so with that, it is my great pleasure to introduce tonight's speaker, Dr. Colin Averill. Usually when you start introductions, the first thing you say is where someone works, and that is actually surprisingly hard to pin down for Colin these days. So, and it is because Colin wears lots and lots of different hats, right? So his academic home is at ETH Zurich, where he's an affiliated scientist in the Crowther lab. Colin's also an academic science advisor to Restore, which is an international nonprofit promoting natural restoration work. He's an advisor and co-founder of SPUN, the Society for the Protection of Underground Networks, which we'll hear more about over the course of the next day or two. And recently, Colin has shifted to focusing almost entirely on founding Funga, which is right here, which is a public benefit corporation that works to leverage scientific information about the fungal microbiome to improve forest restoration and carbon capture initiatives. And that's got the ultimate goal of creating more resilient and carbon-dense forests. So we're in Boston, of course. We love the home team here. And Colin is indeed just that. He did his bachelor's degree at Boston University, and then afterwards he stayed for 2 more years to work as a technician in Adrian Finzi's lab. He then went on to get his PhD at the University of Texas under Christine Hawkes, and then used a highly competitive NOAA Global Change postdoctoral fellowship to come back to BU to do a postdoc with Jenny Botnagar and Mike Dietz, right? All right here. And then he transitioned to Zurich, and since then he has sort of been juggling academic work and nonprofit science application. So Colin only received his PhD in 2015. Despite that, he has an incredibly prolific scientific record. He's published in Nature, Science, PNAS, Global Change, Ecology Letters, and pretty much all of the other relevant, highly respected journals in our fields. He's won a slew of research funding through grants and competitive fellowships. He's given a TED Talk about harnessing fungi to build carbon capture, and he's been highly successful at taking the excellent technical science that he works on into the public domain. So I don't think there is a scientific version of the EGOT, but he's pretty close. He's got he's very proficient in a lot of different fields, which is something I really admire about Colin. I personally have spent my early career closely following Colin's work, largely as sort of a bellwether for where a lot of the sort of forefront of our particular field is moving. And I really appreciate how his science is doing that for us. The one thing that I think I admire most, though, about Colin's work is that he is really he's got a drive that tries to move scientific information into the field of public domain to try to actually take action on improving the forests that we all study, right? And I think a lot of researchers in our field, they want to branch out from just writing scientific papers to putting more actionable more actionable items in the repertoire, but often we don't have the initiative or the courage to do that, and that's something that I really appreciate about what Colin does. So he's a great example of how you can connect really intense, high-level scientific inquiry and the public domain to both understand and create more resilient and better managed forest ecosystems. And I think that's something that I really like about Colin's work, and I think that is a lot of what he's going to talk about this evening. So with that, I will shut up and let Colin tell us about restoring the forest fungal microbiome. Please help me in join me in welcoming Colin.
Thanks so much for the introduction, Ben. I do not deserve it. So thank you all for being here. My team really focuses at the intersection of forest microbiology, forest ecology, carbon cycle science, and natural climate solutions. And so we try and bring all those fields together to discover new things that can help us take action on biodiversity and climate crises. But before we get there, I think it's really important to start at the top to understand how fungi can be so important to the functioning of forest ecosystems and at the same time so often overlooked. And I think part of that is because most of what we know about forests is based on things we can measure above ground. So historically, an ecologist would come to this place and they'd count the number of tree stems they'd find. They'd identify which tree species they are, and they might even today probably remotely sense features of this forest canopy from space. Ugh. And all of that absolutely makes sense. You know, above ground is where photosynthesis happens. This is how carbon and energy enter ecosystems. This is how forests absorb CO2 and can buffer the planet against climate change. However, we also know most forests are limited in some really meaningful way by soil resources like water and nutrients. And to get those, trees have to build roots, and trees build a lot of roots. So in some forests, there can be as much or more biomass below ground and root structures as above ground in stems and leaves. And these root networks are massive. The root networks of adjacent trees are usually intimately intertwined. And so as you look at root architecture, it becomes very clear very quickly that there's this enormous potential for important ecological interactions to happen below ground. However, when you look closely at roots, and I mean very closely, like you'll need a microscope closely, you realize they're not really roots at all. They're also fungi. So most trees on Earth form a biological partnership or a symbiosis with mycorrhizal fungi. This, in my opinion, is one of the most remarkable images ever captured of these fungi. It's a confocal microscopy image taken by Vasilis Kokoris, a professor at the Free Institute of Amsterdam. In the background, you can see this sort of dense network of fungal hyphae in the corner. These are like roots, but for fungi instead of plants. And in the foreground, you can see this wild, multinucleated fungal spore, which looks totally unreal, but absolutely is. This is the reproductive structure of the fungus. It has the potential to become an entirely new fungal network. And what's going on here is that the plant and the fungus are trading. And so the plant is taking some of that photosynthetic carbon it captures above ground and it's feeding it to the fungus, which in return is passing back soil resources like water and nutrients. And I want to emphasize that this symbiosis is really essential to plant biology. There's actually evidence when plants first made the evolutionary transition from living in water to living on land, they started to evolve this symbiosis before they even evolved roots. However, much of how we know these organisms are so essential to plant biology is from experiments that often look something a little bit like this. We like to call them sometimes big plant, little plant experiments. And so the big plant was grown with its mycorrhizal fungus, and the little plant was grown without a mycorrhizal fungus, and the big plant is clearly bigger. And it's growing more vigorously, and other experiments might show that it's more resistant to pathogen attack or drought. And the magnitude of those effects probably vary based on the particular species of fungus or the environmental conditions or soil nutrition and stuff like this. There's so much context dependency. However, what we can learn from experiments like this is still fundamentally limited, and that's because a seedling is not a forest. And in a forest, we never really have this sort of mycorrhizal, non-mycorrhizal case. Actually, there's always fungi present in the forest. There's usually dozens to hundreds of mycorrhizal taxa coexisting on the root network of a single tree. The question isn't what happens with or without mycorrhizal fungi. The question is, what does incredible biodiversity of these mycorrhizal fungi really mean for how entire forests function? Or put another way, I like to cast it this way. What does a healthy forest microbiome look like? And I realize the word healthy is squishy. I think it's usefully squishy, so bear with me for a moment. We're far closer to answering a question like this for people than we are plants. And the Human Microbiome Project has really led here. And so your body is a microbial ecosystem. Each of us houses an incredibly biodiverse community of bacteria within our gut, and we now know this has a profound effect on our health. And this was first discovered by medical microbiologists using DNA sequencing to characterize which bacteria live in hundreds of people's bodies. But importantly, for those same individuals, they collected paired health information. So they asked them, are you sick? If so, what do you have? What's your blood pressure, your digestive health, your mental health? By combining all of that, they could begin to identify combinations of bacteria that are linked to health and disease states. And furthermore, they could use those models as essentially a roadmap to guide microbiome transplant therapies, fecal transplants, essentially ecosystem restoration, but for your body's microbiome. And these types of transplants are the treatment for antibiotic resistant C. diff infection. And there's a huge slew of diseases that people are investigating actually treating this way. And so my team at ETH Zurich's Crowther lab started to ask, what would it look like to take this Human Microbiome Project approach, but apply it to the forest? Could we identify combinations of fungi and bacteria linked to forest health outcomes? And we're particularly focused on the rate at which trees grow and pull carbon out of the atmosphere. And so that's what we've spent the last 5 years doing. And so these next two papers I'm going to talk about were done with Mark Anthony, who's a postdoc who worked with me at ETH Zurich. He's now a professor at the University of Vienna. And what Mark and I did was we teamed up with a forest monitoring network called ICP Forests. And so this is a European-wide forest monitoring network. In each of these locations, foresters have been monitoring forest health outcomes for decades. So tree growth rates, survival rates, crown conditions. Again, we really focused on how fast these trees are putting on wood and locking up carbon in wood. In each of these locations, we asked those foresters to go out and collect a soil sample, which they then ship back to our lab at ETH Zurich so we could extract DNA and sequence it to characterize which fungi and which bacteria live in each one of those locations. And then we built models of forest growth as a function of all the things we usually put in models of forest growth. So tree species type, how old the forest is, stem density, climate, aspects of soil like soil pH and texture. But then we also included in those models covariates that we derive from those microbiome community tables. And this is what we found. So here I'm plotting tree growth rate on a log scale as a function of fungal community composition, here represented as the first principal component of the community matrix. You don't need to know what that is. You really just need to know this is a metric trying to capture a lot of the variation in those fungal communities. The closer together 2 points are on this x-axis, the more similar their community is. The further away, the more different. We find which fungi are inhabiting this forest is linked to threefold variation in how fast these trees are growing and how quickly they're pulling carbon out of the atmosphere. And I want to emphasize again that this is the effect size we see after we control for age and climate and all these other confounding factors that we know also drive tree growth. But we also wanted to know a little more here. We didn't want to just know whether or not fungi predicted tree growth. We wanted to understand if we could drill into this community information, understand what are the members that drive this effect. And so here I'm plotting the standardized model coefficient, which is basically how strong that effect is across a bunch of different models. And what we did is we took that fungal community information, and then we subsetted that table to say, okay, give me only the mycorrhizal taxa, the ectomycorrhizal taxa in this case, or the endophytic fungi, which is another class of fungal symbionts, or the saprotrophic fungi, these are the things that decompose organic matter in soils, or the pathogenic fungi, the things that kill plants. And this is what we found. Basically, using all of the fungi, the entire fungal community table is always best, right? That's the best way to predict how fast these trees are growing. Maybe this isn't surprising. There's fundamentally more information in the entire community table. And also we know, all of these fungal groups are actually interacting and they all play important roles. And so if you include information about all of them, perhaps you can do better. But after that, we saw these two key groups of fungal symbionts pop out as significant predictors. So the ectomycorrhizal fungi, again, these are the fungi I focus on that form a symbiosis with the tree roots. And endophytic fungi, this is another class of plant beneficial fungi. They can live inside roots, but they actually don't form this structure like mycorrhizas. They can also live inside leaves. They live in between the cells in the leaf. Some leaves can have over 100 species of endophytic fungal taxa in them. They're just super cool groups of fungi. So we see both these symbiotic groups are strong, seem to explain a lot of the effect we see in the fungal community. And interestingly, we don't see major signal in the decomposers, the saprotrophs, or in the plant pathogens. We can go even a little bit deeper here as well in the ectomycorrhizal taxa because they're a particularly well-studied group and they have a lot of sequenced genomes available. And so this is actually a different paper Mark and I did. It should have a different reference here. That's my fault. I'm sorry. But basically what we did is we took our fungal community table, so our description of which fungi are living in the soil, and then for each of them we asked if there was an annotated set of fungal genomes in the Department of Energy Joint Genome Institute's genome library. And so that would tell you things like this fungus has this many copies of manganese peroxidase 7, or this many uptake transporters for ammonium, things like that. And then finally, not every fungus is in that database, but actually a lot of these genomic traits have strong phylogenetic signal. And so we used phylogenetic models to sort of gap fill. And we go into way too much detail about this in the paper, and you can learn more about it there. But basically, we turn this description of who is there into a description of what they can do from a functional genetic perspective. And this is what we find. Oh, wow, that's really bad. It wasn't like that on my computer. Whatever. It still isn't like that on my computer. Tree growth rate. So basically, here we're plotting tree growth rate again. On the x-axis, this says energy and nutrient metabolism genes. We see forests dominated by communities of fungi that invest comparatively more in these energy and nutrient metabolism genes. They grow a lot faster. And actually, a lot of these genes are genes used to take up inorganic nitrogen sources. So this is ammonium and nitrate. These two compounds are the compounds in nitrogen fertilizers that we pour on plants around the world. If you, as a forest, have fungi that invest more in these nutrient uptake transporters, we see that that's correlated with you growing more. Conversely, if we look at organic nitrogen cycling genes, forests that harbor fungal communities that invest comparatively more in organic nitrogen acquisition, so actually doing the hard work of producing oxidative, proteolytic, chitinolytic enzymes, breaking apart organic matter so they can take up small polypeptides and amino acids, that's associated with trees growing slower, and that makes a lot of sense. It's a lot more work to do that, right? Inorganic nitrogen is a byproduct of other organisms doing that work. And so this difference in the nutrient foraging strategies of the mycorrhizal communities makes a lot of sense here. And so indicators of this fast growth strategy were actually a lot of taxa within the genera Rusula and Lacaria. Indicators of the slow growth strategy are a lot of taxa in Cortinarius and Nocebi. And so to wrap things up, what we found in this large correlational study is that fungal predictors explain about threefold variation in tree growth rates and the rate at which they're pulling carbon out of the atmosphere and putting it into wood. And furthermore, symbiotic fungi, and particularly ones with specific nutrient uptake strategies, seem to explain some of this correlation. And so there seems to be some intuitive ecological signal, at least in this large correlational data set. And that finally, the most important predictors in these models are like which tree species you are, how old you are, and how many trees per acre are on this landscape. But after that, it's the fungi. And the fungi outrank climate and other soil predictors. They're really strong. They're one of the best environmental predictors of how fast these trees grow. And so the lingering question here, though, is obviously like, are any of these relationships causal? This is a correlational analysis. And so, Does this mean that these particular combinations of fungi, do they actually cause trees to grow faster through a variety of ecological mechanisms? Or have we just discovered a really great bioindicator of site fertility? And another way I think you could phrase this question is, if it's causal, can we manage the forest fungal microbiome to create outcomes? And you might care about that in two particular settings. The first we think about a lot is restoration. If instead of just planting a tree, I planted a tree and its associated fungal microbiome, could I enable or accelerate forest restoration outcomes in a way that pulls more carbon dioxide out of the atmosphere as a natural climate solution? Or forestry for timber is a major use of agriculture across the globe. We know clear cuts actually eliminate about 75% of the mycorrhizal species that form symbioses with those trees. It's based on studies done in Scandinavia and Canada. What if when we're replanting those forests, we also replanted the associated fungal microbiome? Could we rebuild the lost fungal biodiversity in those forests in a way that also creates outcomes for how fast the trees grow and essentially a natural climate solution? I think the answers to these questions are absolutely yes, and I think we've actually known that for an extremely long time. What we don't know is how to do that in a predictable, reliable way. And so I'm going to walk through some of my favorite evidence here and where I see the next phase of this research going. I say grow too much. So this is one of my favorite papers in this area. It's by Alfred Hatch. It was published in 1936 in the Journal of Forestry while he was a scientist working at the Arnold Arboretum in Jamaica Plain, where we're going to be tomorrow. So this is your guide. This is your science. I'm sure you've already read it. And I love this paper for a lot of reasons. One of the reasons is because the first sentence is, this paper has large implications. Let's go. And so in this paper, Alfred basically describes a lot of efforts at the time to plant. They were trying to grow pine trees in the prairie, in the American Midwest, where they're not naturally from. I know that's probably a biodiversity nightmare. Put that aside for just a moment because this is cool science. They couldn't get them to grow. And then he learned that colonial governments around the world were trying to establish pine plantations in Australia, in Southeast Asia, in Africa, and they couldn't get them to establish. And it wasn't pathogens, and they could fertilize them as much as they liked. They couldn't get them to grow. Alfred essentially did these experiments to show that if he took soil from a native pine habitat, he could get them to grow. And he could do other experiments where he could isolate the fungi associating with those pines, the mycorrhizal fungi, and actually recapitulate those responses with pure cultures. And I love this example because basically what they did at this point is they transformed a landscape from one that could not grow trees to one that could grow trees, right? And that is an extremely clear microbiome, ecosystem function, carbon cycle science outcome. I can't think of a clearer one. And then this became standard practice in tree nurseries around the world. They had to get soil because it had spores to inoculate the nursery beds so that they could actually outplant trees that could have their symbionts and so they could grow. So this is an old example. There's analogous examples of this for rhizobia and soybeans in the 1920s, I believe. And we are rediscovering this kind of approach in restoration ecology today. So I'm going to walk through three of my favorite examples here. The headline is the outcome that I think is cool. It is not the title of the paper. This is a study by Jasper Wubbs, published in 2016, his group at NEO, trying to restore northern latitude peatlands in the Netherlands. And basically, they can't get these ecosystems restored unless they actually do these soil transplants from the native ecosystem while they're seeding in the appropriate species. And actually, they can track soil biodiversity outcomes in these restoration experiments as well. They have decades of data from this particular one. And they can show not only are the grasses recovering to the reference community composition, but so are the nematodes, and so are the other microbial groups in the system. This is a study by Liz Koziol and Jim Beaver working in tallgrass prairie systems in the American Midwest, so trying to undo what Alfred Hatch did. Basically, they find if they take soil from an intact prairie remnant, that can massively accelerate the growth and the establishment of mid and late successional tall grass species. They've actually pulled out the arbuscular mycorrhizal spores of those soil transplants and shown they can actually recapitulate a lot of that effect with the mycorrhizas themselves, which is really interesting. This is a project in Wales in the United Kingdom I work on with Dr. Bonnie Waring at Imperial College and a charity there called the Carbon Community. In the UK, there's something called the Woodland Carbon Code. Basically, the UK government is subsidizing the establishment of native broadleaf woodlands on ex-sheep pastures for carbon. So we basically do woodland restoration at the site, or we do woodland restoration and a soil transplant from a nearby ancient woodland. The trees grow faster, they remove more carbon if you co-introduce their microbes. And so I love these examples. I think they're all super straightforward. The first two are especially well done. But they are anecdotes, right? We want more systematic evidence here, and so we did a meta-analysis. We tracked down 80 studies from around the globe in very different ecosystem types where people had done a soil transplant or some other type of native microbial restoration. And we found, on average, the plant biomass response to those wild inoculations was about 64%, with remarkable variation, lots of zeros. The people have done these analogous meta-analyses looking at commercial fungal inoculants in agriculture and in restoration. The average response is 0, so there's something very different about wild microbiome restoration that we have not been able to recapitulate with whatever's in commercial inoculants that are on the shelf today. But I think this wild variation is really where we're at today. Sometimes you do this and it absolutely works and it's visible, right? Like you can visibly see the difference among the treatments, and sometimes it's a wash. And we don't have that predictability right now. And I think the reason we don't is because we don't know what a good donor environment is, right? And so where are you taking those microbes from? And how would you know if they'd ever be good at actually accelerating a restoration outcome or something? And so, how do we identify the right donor ecosystems for microbial microbial restoration? These are the places you would actually take the soil for a soil transplant or some other way you do this. And this is my dream, platform for doing this. You would you'd focus on a particular plant species, and you generate paired soil microbiome data and plant health data across its entire range. And so plant health data could be growth rates, survival rates, carbon capture rates, and you'd build models analogous to the ones we did in Europe that say, these are the candidate donor forests or donor ecosystems that harbor microorganisms that are linked to the health outcome I'm trying to induce in this recipient ecosystem. Then I'd love to screen all of those candidate donors in a greenhouse where you can run really high throughput experiments and make sure they actually establish, right? Just because something is a promising donor, it doesn't mean it's going to live through your transplant approach, right? Like you have to dig them up. Some microorganisms can absolutely do that, some cannot. And so you can trial them in the greenhouse, see if they elicit the plant health outcome you're looking for, see if they establish in the soil. And then that's how you conserve your precious resources to run field trials. You run distributed field trials, and then you iterate on this. And so this is sometimes called the forecast analysis cycle. I was taught this by Dr. Michael Dietz, who's sitting right there. And the idea is that every time you do that, you build models, you forecast what will happen, you do the thing, and then you iterate, right? You get better at doing that every single time. And every single time you do that, you could also spin off inoculant products. And so you might be working with a restoration group who's trying to restore a different part of the world, and you could say, okay, for this ecoregion, here's our best guess at what's good for here, and if you're doing it in that ecoregion, here's our best shot at what's good here. We tried to do this in the Crowther lab, and we quickly learned that this is like logistically wild and is not something you can do in an academic lab. It's really hard to run like 20 distributed field experiments and time all these soil inoculations and operate at this kind of scale. And we realized if we're going to do this, we really need a different thing. We need something and different skill sets than just scientists to actually accomplish like this. And so we founded a startup, and it's called Funga. And Funga exists to run that playbook. And and we're a public benefit corporation. Our mission is to reintroduce fungal biodiversity to forests to create outcomes for climate as a natural climate solution. And again, we run that playbook. Part of that playbook is picking a focal tree species, one ideally where you can put up carbon outcomes quickly and outcomes for other economic things. We've chosen to focus on loblolly pine, also sometimes called southern pine. This is North America's largest timber market. It grows from Virginia down to the northern part of Florida and as far west as the eastern part of Texas. We chose this species for a few different reasons, but the first reason is because it's a native tree species to the south where it's grown for timber. And so there's actually a lot of native soil biodiversity to work with here. Second, because the infrastructure is massive, and we don't have to solve all the challenges a restoration project has to solve because they have massive tree nurseries. We think there's something between 30 and 50 million planted acres of pine forestry in the south. 1.3 million acres are harvested and get replanted every single year. And so that's your total, potential market you could address here, right? How many acres on an annual basis? And lastly, timber is this really important agricultural commodity in the United States and in the world with really big climate impact. So for example, if we're going to decarbonize the built environment, it means we need to build more with wood and less with concrete and steel, which have really hard to abate emissions. The last reason we chose this place, though, is because it's not all about donors. The microbial restoration works when you have a good recipient, when soils are degraded. And we think these ones might be degraded. This is what a 500-acre clear cut looks like in southern Georgia. This is actually a site we work on. And they do a lot of things here. So they first clear cut the trees. And again, studies from different parts of the world show lots of mycorrhizal species cannot survive this scale of clear cut between when they're cut and when they're replanted. But they don't stop there. So after they clear cut, they then burn it. They want to remove all that debris. Then they'll apply glyphosate, often from a helicopter. So this is also known as Roundup. It's an herbicide because they want to kill all of the competing vegetation. And then you'll notice there's actually the slight undulation to the terrain because they plow it and they plant the trees into these mounds. It's like a drainage feature. And we know plowing like that has really degrades soil biodiversity, especially fungi. And so I think, when we see all of those together, Dr. Ian Ware on our team calls that the full nuke. And when we see the full nuke, right, in all its glory, it's easy to imagine how soil microbial biodiversity might be degraded in this system and how active reintroduction of organisms might actually do something meaningful here and cause an outcome. And so that's what we do. We run this playbook, right, the one we've already walked through. And so this is the geographic range of southern pine in pink. We have sourced soil samples from 500 locations already across the south. So in each of these locations, it's just like Europe. We have a soil sample, and we also have a diameter survey of the trees. We know the day they were planted. We know lots of things about management, tree survival, many other things. We bring in climate and soils data. We build models to essentially put forward candidate donor ecosystems. We identify forests in the south that are still holding on to some fungal biodiversity that at least correlationally is linked to the outcomes we want to see. And again, we see similar correlations in the south and in pine as we did in Europe. We then bring them into the greenhouse for two reasons. The first is, let's do a screen, make sure these microbes establish, elicit a growth response. Let's also make sure we don't cause any unintended consequences. And then once they've gone through that screen, they go into field trials. And so we currently have field trials running in these 20 locations, and every single place is a place where we have a block randomized controlled trial where we have trees we didn't inoculate or ones that we did. In most of these locations, we're testing multiple flavors of inoculation, and so there's, I think, something like over 50 experimental contrasts in the field. What I can say today is we only have data back for a few of these, right? We've only been a company for less than 2 years, but 89% of the inoculants we've tested to date have created a significant positive outcome for either tree growth or tree survival in the field. And the point here is that then we can spin off a product, right? And so when I say product, I want to emphasize we're not trying to apply the same group of microorganisms everywhere. They're regional specific. We have different flavors for the Western Gulf and the coastal plain and the Piedmont, and they get more specific as we get better. But we actually already did this on 1,900 acres of timberland this past winter, which is the planting season. And the idea is that we're going to continue to sample. That's going to continue to prove, how well we can put up outcomes in the field, and we're going to continue to run the forecast and analysis cycle to make this better. And I can share with the other thing that's really important, though, is that we're actually causing this for the reason we think we're causing it, that we're actually shifting biodiversity below ground. And so here I'm showing basically an NMDS ordination of fungal community composition. And so this is basically a two-dimensional representation of biodiversity. If you're not familiar with them, I think that's enough to understand them. And you can think of this as biodiversity space. The closer together 2 points are, the more similar they are. You'll see that these are just industry standard seedlings from the tree nursery that we sequence. And you can see they live in this very confined region of biodiversity space. And these are the same seedlings grown from the same tree nursery that live on the same tables, but we've inoculated them with our different donor soils. And we've achieved this outcome where we've massively expanded this space in which these seedlings occupy. And so this is what we would expect to happen. We are introducing fungi and showing up in our sequence data. However, this is actually the ordination when you put in the donor forest, right? So these are the places we took the soil from that we used as an inoculant over here, and they are very far away, right? And this is also expected, right? We're not expecting to recapitulate like an intact forest microbiome in a day, but you can actually show that the inoculated seedlings are more similar to those donors than the control seedlings. And furthermore, this is really interesting. If you look at alpha diversity, you see it's lowest in those control seedlings, it's highest in the donor forests, and actually it's nearly the same in the inoculated seedlings. And so a lot of the difference between those communities is not necessarily because the species are absent, but because the representation of the different species in the community hasn't shooken out yet. We wouldn't expect it to in a year. But the point is, we've set these seedlings on a trajectory towards there, right? Like we've accelerated the dispersal and establishment of these growth-promoting fungi, and this is what we believe causally underlies the changes in tree growth and tree survival that we're seeing. And so the last thing I want to say here about fungi and the work we're doing in pine is that it's a startup for a reason, and that's because scale is really important. Taking this from a cool set of science studies that we've seen and has some promise to a technology that's actually reproducible and predictable requires just a massive amount more of replication. We just need to do this more. Climate crisis is today. Biodiversity crisis is today. We also need to do this fast, and that means we need to go to scale. I'm going to tell you the scale story of the IFCO PRT tree nursery in Moultrie, Georgia. So this is the biggest seedling grower in North America. This is just one of their tree nurseries. This is what it looks like. This is where we do the production scale inoculations. And I think they grow either 60 or 80 million seedlings just at this one location. And this is part of how we can take things like, that are quote unquote low tech, like soil transplants, like literally throwing dirt in a hole, and bring them to massive scale, right? Because an acre of pine forestry starts on less than a square meter of nursery footprint. And we're actually developing ways such that, we don't ever have to take any soil out of the forest again. And so there's new ways we're working with these things, and basically solving those kinds of scaling problems isn't always exciting, ecology, right, that'll get published in a high-impact journal, but it's really important technology problems. And we get to work on those as well, which makes me really satisfied because we need to bring this to scale. And also, I believe that bringing this to scale is the only way we're ever going to have the replication necessary to really understand the predictability of all of this. And this doesn't work without an incredible team. I'm super proud of these 13 people, and I think we're making a job offer this week to a 14th. Half of them are ecologists that come from our community, from places like the US Forest Service, NASA's Terrestrial Carbon Cycle Science programs, The other half of them, some of them are foresters. We have actually, that's the person we're hiring this week, and Brian Box on this slide is a working U.S. forester in the South who helps us get stuff done on the ground. And the rest of the half is a business team, right? Like to make this scale quickly, we need to build something that people will pay for and show traction such that we can attract investment so we can continue to grow this and achieve these kinds of scales. And so they make sure we're building products that people want. They make sure that our message is sharp. And they make sure we manage relationships with landowners, with the major forestry companies in the United States. They're essential. And so this multidisciplinary team, you can't really do it without them. This is the hard thing to do in academia that startups are built for. So I'm coming to the end of the talk, and I want to close out on this question that I started with. What does a healthy forest microbiome look like? Or a prairie ecosystem, or a mangrove, or an agro-ecosystem, right? I love this question because it's not actually specific to forests. It generalizes across ecosystems. And in reality, this isn't even specific to microbiology. This is a question ecologists have been asking for a very long time. How does biodiversity shape ecosystem function? But when we present it in this way, it begs applied questions. It begs question of how do you apply biodiversity as biotechnology? And I think that's really exciting to me because the history of biotechnology has really been this exercise in reductionism. And let me use agriculture as an example, right? In agriculture, we identify high-performing plant species and then strains of those species, and we selectively breed them, and now we genetically modify them, and then finally, we put a small handful of genotypes out in a vast monoculture, as far as you can see. And to be clear, this has absolutely worked, right? Corn is like 7 times more productive than it was in the 1890s. Pine tree is about 427%, because I think about pine trees too much. But we also know we built ecosystems that are increasingly fragile. So they're increasingly sensitive to extreme events and novel pathogens, and they're incredibly reliant on chemical inputs we're now coming to understand have really serious unintended consequences. We now have the data, the mathematics, and the frameworks to start going the other way, to lean into biodiversity and complexity. And once you start, the question really becomes, by building biodiversity back into these systems, can we better deliver yields and carbon capture and all the other services we ask of these ecosystems, right? And so I think the exciting thing here is that the next frontier in biotechnology may actually just be biodiversity as biotechnology, and I think it really needs to be. We are living through a global mass extinction event. And as we ask more and more of our natural and our managed landscapes, we also need to be asking how they can best support the remaining biodiversity on this planet. And when we ask that question, we can't just think of the plants and the animals. We also need to think of the majority of species on this earth that are microbial. We need to think about the yeast and the mold and the mushrooms. We need to think about microbial biodiversity. And so with that, I want to say thank you very much, acknowledge a ton of funding from the academic community for the science program at ETH Zurich and the Crowther Lab, from our venture capital investors at Funga, and I'd be happy to take any questions. Sure.
Yeah, so thank you for the talk. This is really interesting. At the beginning, you showed a plot relating the functions of the fungal microbiome to the rate of growth, where you were saying like chitinases, et cetera, and then like inorganic nitrogen. How does that track with like the carbon-nitrogen ratio in those forest ecosystems?
I don't totally recall all the covariance of all the covariates in that study. I do know that we fit a subset of models with carbon and nitrogen ratios in them, so we thought a lot about that in pH, but I couldn't tell you off the top of my head.
I guess my expectation would be that in carbon, like really high carbon environments, you'd have more of this kind of like chitinolytic activity compared to the sort of inorganic nitrogen transport, and that would just drive that correlation. But
Yeah, so you could imagine, right, that the carbon and nitrogen ratio is actually underlying that balance of microbes, right? So the more organic nitrogen communities are in the higher C to N ratios, and so the fungi are acting as a bioproxy of really low nutrient availability in those systems. I think the other side of that that's really interesting, though, is that we know some of those same fungal species can induce and aggravate nutrient limitation, and so it's not actually obvious that one causes the other, right? Because we know those things are moving together and feeding back on one another. Jenny.
This is great.
Thanks.
I paid attention the whole time.
I believe you.
It's a miracle. No, it's really okay. When would you output your seeds? So do you direct seed in the location that you want them to grow really big? How does the growth and transplant process? Is there any transplanting of your seedlings when you output them, or are they just growing where you want a new stand to be?
Yeah, so it depends on the system. So when we do our work in Wales, we follow the best practices of people who work there, and generally it's plant in the winter during the planting season. There we straight up, we have our donor soil, and it's like about 100 mils, which looks like a small fistful of soil in the hole, dirt in the hole. Love it. With the pine trees, it's dirt in a hole does not scale past like 100 acres. It's insane. It's very heavy. In the pine trees, we actually inoculate them in the tree nursery, and so we use very, very small quantities of soil, and now we're experimenting with approaches where we make soil in our donor forests, and so we bring compost and other substrates we're testing as something that we can actually put out, get colonized, and then harvest and replace.
Well, the reason I ask is because you'll get succession of time. And when I saw that field of seedlings without any mature trees that are close by, potentially, I was getting concerned that you might be inoculating and establishing like a young seedling ecosystem, but they might not have access ultimately to later stage fungi. And especially when you showed your NDS and you see that forest fungal community way off different from the other ones, I wonder if that's because they're mature trees and they're just in a very different community than they're mature. So I wonder if you guys have thought about how to get those later stage fungi on the trees that you're trying to grow.
I think that is really a lot of our goal, right? Accelerate the dispersal of organisms that may have got there eventually, but might not have gotten there for a decade in a way that actually accelerates growth. And, it's important to remember that the counterfactual is those seedlings on a table, the ones we just don't inoculate. That is what's happening in pine forestry at massive scale today. And there are some fungi in there. They're the ones you would expect. They're Telephora and Sinococcum, these really weedy species. But yeah, that's the goal. That's what we're trying to do.
Well,
and
you could leave one tree, like a big tree, like growing in the middle, so right? So just to get those really late stagers growing in that space.
Yeah, and I thought about this a lot too in the context of how does a seed normally meet its microbiome, right? Like you could think in the simplistic case of a seed falls from a tree and it falls in the soil and that's what it needs, right? That's why I think the soil transplant actually works very well. I've met groups that are actually starting their seedlings in soil around mature trees in Spain, trying to do this, which I thought was really interesting. And you could imagine having a nurse tree in the system to do that. There's also people who are doing selective logging. The other thing you could do is not destroy it in the first place, not do the full nuke, right? And you could leave retention trees, which is something Susamart actually works on in British Columbia. Jennifer.
In Mark's paper, when you showed that plot of the different functional communities and growth, I didn't say AMs in there. Are they part of the endo group? Or are they not in that plot?
So they're not in that plot for a few reasons. So first, most of Europe is 4 tree species, and all of them are ectomycorrhizal. And so that's what we focused on. And then, of course, we're also using ITS sequencing, which, as I'm sure you know, biases against the mycorrhizal fungi. So we could do this though, and we are working with the Forest Geo Network to do sequencing in all those sites, and we're doing both ITS primers and AMF primers to try and ask these questions there.
Okay. Just thinking, when you get beyond Loblolly and Pines River, you're up in Oaks, and you want to move into the products, other places that also need foresting. I think that names are going to need to be there as well. And I would be shocked if there's a different pattern, but since I better go measure it, it better be you.
As soon as we can. Adriana.
You are sequencing the soils, right? I was curious if you have identified certain taxa that are better than others, for example.
So we do see certain indicators. Species pop out in these analyses, and so often, for example, indicators of like slower growth in Europe are species in the genera Cortinarius and Anosabe. Indicators of faster growth are often in the genera Lacaria and Russula. But we generally find that no individual species is a strong predictor on its own. They have signal. The best predictors are usually the variables we derive from the entire community.
Let's go, Joe.
Let's go, Joe.
All right, so my question, I guess, builds off Jenny's a little bit. So there's a lot of research on the impacts of different silvicultural treatments on wood growth, right? Has a lot been done on the impacts of different silvicultural treatments on microbial abundance and diversity? Like I'm thinking, if you have a shelter wood treatment where you have a few trees remaining, what's the impact of what the microbial community looks like? I guess this would be very much against the idea of inoculating soil, so maybe bad for the company. But I'm curious what it looks like in general for the microbial community under various silvicultural applications.
I would say there's actually a lot of studies out there that have looked at the effect of some silvicultural treatment in a handful of sites in Eastern Oregon or in Idaho or in North Carolina. And what's really, and there are effects, there's often effects of different management practices. What is generally missing, however, is anything that's synthetic at a large scale. And so I think we don't know with any consistency, across a wide set of sites, what's normal.
Colin.
He doesn't know my name, so I wasn't sure if he would call me. Thank you. I'm curious about all the rest of the microbial world, including the 10 to the 9th bacteria per gram of soil. And are they coming with your soil, or is it sterile? Are they affecting nitrogen cycling? I mean, we know they are, but I'm just curious how it figures into your company.
Oh yeah.
Inoculation.
So I'll back up to Europe. We also sequenced bacteria in all those soils using 16S markers. Remarkably, we could not find signals in the bacterial community that predicted tree growth. And so we thought that was interesting in and of itself, that there's remarkable signal in the fungi and not the bacteria. When we do these transplants, we do them as whole microbiome transplants, whole soil transplants. So fungi are coming along with bacteria, are coming along with archaea and native soil viruses and nematodes and all sorts of stuff. We think that's really important. We know, even though we see markers of these things and there are indicator species, generally reduced diversity subsets, adding 1, 5, 10 species doesn't work. People also see this in medical microbiology, right? Like people, know the fecal transplant works for treating C. diff, and people have made reduced communities, and they generally do not work or are not as effective as the fecal transplant. And so it's likely, it's all of that's to say is almost certainly all of that biodiversity is doing something, including the prokaryotes, even if we don't necessarily see it in the sequencing signals.
But so then for your company, would you be selling soil?
We don't sell any inoculants to anyone.
Not now, but I'm assuming this is supposed to be a product in the future.
So we don't sell inoculants ever. We do inoculation for partner forestry companies for free, and in exchange we get a lease that gives us the right to develop a carbon project and market carbon credits.
Ah, okay, thank you.
No problem.
Yeah, so Colin, you spent a lot of time talking about tree growth and survival, and I love tree growth and survival more than most anyone. But that's not the total carbon budget. And so have you looked at how these different microbial communities select for soil carbon storage, and if there's any sort of trade-off between above ground growth and below ground storage.
Yeah, so we looked at this a lot in the recent paper we had in Nature Communications because we actually have total soil carbon storage for the vast majority of those sites. We basically see that tree growth rates and soil carbon stocks in these forests are moving together, which we don't think is super surprising. These are, the vast majority of European forests are managed, they're planted forests. And basically what we see is soil carbon goes down when you clear cut or when you harvest, and it recovers with time, essentially. And so we actually suspect that, the hard thing about soil carbon is there's very few time series of soil carbon. So all we can do is infer from patterns we see in stocks. But basically we see that older forests have more carbon, right? There's a time thing. We also see forests that are growing faster seem to have accumulated carbon more quickly. And so in the Nature Communications study, the microbiome's moving, it has a positive association with above ground carbon and below ground carbon. They're moving together.
In the very beginning of the talk, you mentioned about symbiosis between photosynthetic trees and fungus. There are at least about 8 known species in flowering plants which are non-photosynthetic. One example is monotropon, and it is surviving just because it is fed by the fungus. Do you call it as parasitism, or does fungus get anything out of plants like monotropon?
I think that's an excellent question. Yeah, so you're talking about mycoheterotrophic plants, right? Like non-photosynthetic plants that are essentially, they get all of their carbon from other trees and they get it through their mycorrhizal symbiosis. It's some of the strongest evidence for common mycorrhizal networks being important flows of carbon to plants. I have no idea the answer to the question, but I wouldn't, I think they're often assumed to be parasitic, right? But you could also imagine it could confer all sorts of positive traits as well. It depends on your scope of your investigation. So thank you for just bringing them up because I think they're fascinating.
Let's go one more.
That's fine.
Yeah, I was curious on your initial analysis of where to get soil from, right? Like you were studying tree growth. What's happening with the trees that don't have mycorrhizal association in those forests that you diagnose as potential problems? They are also growing intensely, or definitely you're just studying the mycorrhizal species? How did you do that analysis?
Yeah, so everywhere we've done this correlational work, we've been focused on ectomycorrhizal tree species. And so we haven't been to a place that has non-mycorrhizal trees. You can imagine non-mycorrhizal trees also still have an associated microbiome that's specific to them, and that these efforts may translate that way. I tend to focus on the ectomycorrhizal trees just because I know, especially pines, are super dependent on these mycorrhizal fungi. I've tried to do root tip colonization analyses in pine forests, and it's a dumb thing to measure because nearly every root tip is colonized. And so that's one of the reasons we've started here. But I imagine this could extend to other things. And I don't know the mycorrhizal status of soybeans. I bet you do. Okay, yeah. But there's lots of nitrogen-fixing plants too that have rhizobia that aren't necessarily mycorrhizal.
Okay, I'm gonna cut us off there. There's refreshments outside. If you have additional questions for Colin, please stick around and ask them over here. Thanks, everyone.
Thank you so much, Ben.